So in the final section of this lesson, we're going to talk about staining techniques, which are important for enhancing contrast and the ability to see microbes under the microscope. Staining is great because not only does it let us see microbes more clearly, but it also helps in differentiating between different types of microbes, like different types of bacteria. So here we'll discuss the different types of stains and how they are used in medicine as well. So by the end of this specific section, you'll be able to have an understanding of staining, simple stains, negative stains, differential stains, and special stains. Okay, to start off, before you stain, well, what is the purpose of staining cells? The purpose of staining cells is to look at transparent cells, and it's also important in diagnosis. So we can use it to distinguish between different types of bacteria, which you'd give different types of antibiotics depending on what color they stain. So staining is a great technique. It's used all the time in lab in clinical microbiology. So we can do live versus stained specimens. So let's talk about that. So a stain is a chemical. At the end of the day, there's so many different stains, but it is a chemical. So it may be able to harm your organism. So sometimes the reason why people like to not stain is that you can look at natural behavior. Staining is great if you want to see clear visualization of structures. But the con is that sometimes it can kill cells or limit their observation. Before you stain a cell, you always have to what we call fix it on a slide. It means that you have to get it to stick on your slide, because if you don't stick it on your slide, then it's just going to fall off when you do the rinsing part of the stain. I don't want to talk too much about lab because we do have our own lab section and lab slides. But what I want to say is before you add any stain, so imagine this is a slide. Let's use our example of a patient's urine sample. You got a urine sample, you put a few drops on there. I can't just put the stain on it. If I stain it, it's just gonna get removed. So what you have to do is you have to prepare a smear. That's what we say in science. So you put your patient's urine sample, and the way to get it to stick on the stain, it's very easy, is you just pass the slide through a little bit of heat. That's it. And that's called fixing a slide. It's called heat fixing, and it attaches microbes to the slide, and now you're able to stain them. So just wanted to say that making a smear means making your slide, means heat fixing your cells on the slide, which is a fancy word of just put your sample, your blood sample, your urine sample, your saliva sample, and you pass it over heat for a little bit, and then you're ready to stain it. There are so many different types of stains, and here are the types of stains that I want to talk about for this class. So simple stains involve just a single basic dye, making it easier to see the shape and structure of a microbe. Negative stains use acidic dyes that instead of staining the cells, they stain the background. Differential stains like the Gram stain and acid-fast stain differentiate between different types of bacteria, and this is important a lot of times for medical diagnosis. And then special stains target specific structures in bacteria, in microbes, not just bacteria. So we're going to briefly go over each one. Okay, starting with simple stains. So simple stains involve the use of a single basic dye to highlight a microbe, making it easier to look at. It's a basic dye like methylene blue, and it's just basically, it's almost like food coloring. They're quick and they're easy and they're ideal for basic observations. Now, let's say we want to look at something else, or we want to do something more fancy. There's something called a negative stain. So negative staining is a technique where instead of staining the actual cells, you stain the background. And the reason that you do this is because sometimes cells will not take up stains, and I'll talk about why that is. So this method uses acidic dyes like something called India ink, which don't penetrate the cells. So as a result, the cells appear as clear and unstained areas, but you get this nice dark background, which is very useful for looking at the cells. Now, why wouldn't you just stain the cells themselves? And the reason why is because this you're using with special cells that will not take up a stain. So for example, some bacteria have something called a capsule. A capsule is actually almost exactly what it sounds like. Think of like a pill capsule. It's a covering, and it's a gelatinous covering that some bacteria have. And this does not allow a stain to go in. So for bacteria that have capsules, you can try to stain them, but it will be very hard. So instead of staining the bacteria, we stain the background, and that is called negative staining. So it's when we stain the background. Next, we get into differential stains. So differential stains are used to differentiate groups of bacteria. Two examples of differential stains is there's a stain called a Gram stain, and there's a stain called an acid fast. Gram stain differentiates what we call Gram positive from Gram negative bacteria. Acid fast stain tells you if a bacteria is acid fast or not acid fast. Okay, Gram stain. We're going to learn all about Gram stain in this class. This is just an introduction. Gram stain is the go-to stain in microbiology. It's one of the most important staining techniques. It differentiates bacteria into two groups. We call them Gram positive or Gram negative. And if anyone is wondering, the name Gram is not very significant. It's just, it just came from the person who discovered this stain. His name was Gram, his last name. So when we think of bacteria, most bacteria fall into two categories. You will learn about this later on in class. We either classify bacteria as Gram negative or Gram positive. So if you have a bacterial infection, a microbiologist may ask, is the bacteria Gram positive or Gram negative? The reason you'd care is you're going to give different antibiotics for a Gram negative infection than a Gram positive infection. And your patient might even be infected with both, and then you'd give them a different type of antibiotic. So Gram positive bacteria, we'll learn all about them, but their cell structure is they have a lot of this thing called peptidoglycan in their cell wall, and Gram positive bacteria stain purple. So the way I memorize it is positive, lots of peptidoglycan, purple, PPP. That's how I memorize it. Gram negative bacteria have a thin peptidoglycan layer and an outer membrane, and they stain red. So that is the difference. And this is a Gram stain. And the way that a Gram stain is done is you put your patient's sample. Let's say their urine sample. Did they have a Gram positive infection or a Gram negative infection? You put their urine. The first stain you add, the primary stain is crystal violet. And then you add a mordant called iodine. A mordant is just a fancy word for something that helps the initial stain stick even more. So you add iodine, and then you add some alcohol, and then you add a secondary stain, which is safranin. That's also called a counter stain, and you're going to do Gram stains in lab. But anyway, if this is the result of your patient's sample, if I tell you, is this Gram positive or Gram negative, this patient is infected with both. I see Gram negative bacteria, they're red, and I see Gram positive bacteria, they're purple. Same with this patient, they have both. And that's the case a lot of times. But there are a few times when you get a sample from a patient and it's all purple, so it's Gram positive, or it's all red and it's Gram negative. So this is the Gram stain, it's a differential stain. The next differential stain you'll learn about is the acid fast stain. So acid fast is a type of stain that is used for specific types of bacteria. It's used to identify bacteria that have waxy cell walls, which are resistant to the staining in Gram stain. So an example is Mycobacterium bacteria. These cause diseases like tuberculosis, which is a really bad lung infection. They're acid fast because they have a lot of high waxy lipid content in their cell walls, which we would say, we call that mycolic acid. So in this technique, you use the primary stain and you do all the staining steps that we'll talk about. And acid fast bacteria retain the pink color, and non-acid fast bacteria turn blue. Acid fast stain is really important in diagnosing tuberculosis. So this is where we would use that as one example. So the bacteria that causes tuberculosis is called Mycobacterium tuberculosis. If someone is coughing a lot, they could have COVID, they could have tuberculosis, they could have the flu, they could have a lot of things. But let's say that you get a sputum sample, which is almost like a very, very deep saliva sample. You would do an acid fast stain, and if the result is it turns pink, that confirms that they probably have tuberculosis because that's acid fast. And if it doesn't stain pink, it stains blue, then that's non-acid fast. So in this method, you apply your primary stain, carbofuchsin, which penetrates the waxy cell wall. After decolorization with acid alcohol, only acid fast stains stay pink and non-acid fast stains stain blue because you add a counter stain, which is also called a secondary stain that's blue. And then the final slide of our stain lecture is special stains. With special stains, they're used to highlight specific structures in microbes, which can be important for identification of microbes. So capsule stains are a type of negative stains that highlight the basically capsule around some bacteria, which can be a very important diagnosis for disease. So there's something called a capsule stain. It's a type of negative stain. It allows you to see capsules of bacteria. Endospore stains are used to identify bacterial endospores, which are these very resistant structures that some bacteria can make under stress. So the endospore stain tells you if bacteria have made endospores. And then the flagella stain is used to look at flagella. Flagella are structures that some microbes have that give them the ability to move. So that's a lot at this level in the course, but what I want you to know is capsule stain stains something in bacteria called a capsule. Endospore stain stains endospores, and flagella stain stains flagella. And then a future lessons about cells, you'll learn about what a capsule is, endospore is, or flagella is, but there's special stains to stain them. I'm hoping that at the end of this lecture, which had 3 sections, section A was Introduction to Microbiology, section B was on microscopy, and this section, section C, was on staining. You are able to answer these questions. This gives you an idea of what I'm expecting. So for example, of things I'd like you to know that microbes are organisms that are micro. They're too small to be seen with our eye alone. I'd love for you to leave this lecture knowing what a pathogen is, because we'll use this word a lot during class. A pathogen is anything that causes disease or makes you sick. What are the 3 domains of life? The 3 domains of life are bacteria, archaea, and eukarya. Viruses do not fall in any of these because viruses are not alive. What's the main difference between prokaryotes and eukaryotes? Prokaryotes do not have a nucleus. Eukaryotes have a nucleus. Prokaryote or eukaryote, bacteria are prokaryotes, archaea are prokaryotes, fungus, protozoa, and algae are eukaryotes, and viruses are neither because they're not alive. Binomial nomenclature. In binomial nomenclature, each organism is given 2 names, the genus and the species. The genus name is always capitalized and the species name is lowercase. For units of measurement, I'd like you to know that light microscopes use micrometers and bacteria are measured in micrometers. That's why we can use light microscopes to look at bacteria. Smaller than micrometers are nanometers. An example of something that's in nanometers is viruses. They're much smaller. We need electron microscopes to look at them. Then we get to different parts of the microscope. So go through the different parts of the microscope. Magnification and total magnification, know the difference between them. And in terms of why is staining important, staining is important because it enhances our ability to look at microscope. So the steps of making a stain is first you make a smear. You heat fixed cells and types of stains. There's simple stains which use basic dyes. Negative stains stain the background, not the cells. Differential stains allow us to differentiate different groups of bacteria. Examples are gram stain and acid fast. Special stains target specific structures. Capsule stain targets bacterial capsules. Endospore stain targets bacterial endospores, and flagella stain targets bacterial flagella. So please take time looking at these questions. They will help a lot.